Hi, am I live, kids? Sorry I kept you all waiting. Hi, am I live? Can you all hear me? Am I audible?
ఇలా ఎల్లప్పుడూ ఎవరూ గ్లామరస్ గా కనిపించరు.
Hi everyone. So you all can see me. Very good afternoon. How is the Josh? Your exams are coming up. Exams coming up. This whole week we have kept only for all of you term 2 preparations with me. Very good afternoon. Very good afternoon. Are you guys ready? We don't have much time. I want to start. Just for all of those who are new to the channel, quickly let me introduce you to myself. I am Dr. Sindur, NEET Zoology expert here at Vedantu, and I have been teaching, mentoring kids for their NEET exams for over 10 years. Hi everyone. Yes, so I did introduce myself. I'm Dr. Sindur and I have done my BHMS from Dr. D.Y. Patel College in Pune. And let us start without further delay. Okay, if you haven't done so far, please go and like the video, share the video with your friends so we all can learn together. It's a super important preparations for your NEET and also your term 2 exams which are coming up. Have you all seen the whole timetable for this week? Have you all seen the whole timetable for this week? Hi, my Nefarians. Hello. Alright. So all of you, if you've please share this video with all your friends so they all can join us too. And if you haven't done so far, I hope that all of you have already subscribed to this channel. Okay. So shall we begin? Shall we begin? You already shared the schedule. The schedule is there on the channel community. Please find it. Every day we're having a one shot. We're also going to be having sample paper discussions. Okay. Now, what are we going to do? Yes, Pranav, this can absolutely help for NEET. Please stay here. We are going to go paragraph by paragraph, complete NCRT, so that, because I know that biotechnology is a long chapter, it's a big chapter. Alright, finally my notes. Yes, Mihika. So biotechnology being a long and a big chapter, what I will do is I want you to keep your NCRT textbook right open in front of you. Every title that's coming in the NCRT, I will tell you that this is the part that we are focusing on right now. Are you all with me? Are you all with me? Yes or no? Gaming with Pulingo. Good, very good question. It's because all my kids here on the chat box, all my Vedantu batch kids here are here. They will tell you very clearly how much I love teaching, which is why I am on this platform. Yes or no, kids? Alright. So without further delay, let's begin, children. One shot, we're going to try to complete. We are going to complete biotechnology in this one and a half hour. Alright, let's begin. So biotechnology, first of all, there are two core techniques, okay, or two core basics which gave rise to biotechnology, right? One is genetic engineering. One is gonna be genetic engineering. The second one is, what is the second one? Check your textbook. Tell me. I need this session to be interactive. Notes you will be getting. Don't bother about sitting and writing the notes right now. I need your interaction in this session right now. Come on, kids. Yes? Along with genetic engineering, you need to maintain sterile conditions too. Bioprocess engineering. Very good, Shri. So, genetic and bioprocess engineering together are giving rise to biotechnology. Now, when we talk about this, the techniques of genetic engineering which are going to be helpful are creating a recombinant DNA, okay, gene cloning and gene transfer. These are the three techniques of genetic engineering which are going to help in biotechnology. Okay, you will get the notes in the Telegram channel. Don't worry about the notes at all. Okay, immediately after the session, I will post it on the Telegram channel. And for all my kids from my long-term sessions, I have managed to almost complete this, so you will be getting further notes too. Okay, now what do we want to do when we talk about biotechnology? If at all you have a DNA that you want, you like it, okay, why do you like it? It's very useful to you. Okay, so that DNA, if you just want to put it into a host which is going to multiply very rapidly, okay, you want that DNA to be produced in excess amount. You want a lot of that DNA, correct? So now how are you going to multiply that DNA? You will take that DNA and you will put it into some bacteria which is able to multiply very rapidly. But if you just take this small DNA and you put it into a bacteria or into a cell which will divide, will that DNA also divide? No, it won't. This cell will consider it as foreign and it will not do anything about it. So there will be no replication. So what do we need to do? This alien DNA, the foreign DNA, okay, needs to be inserted inside the nucleus, inside the genome of the host, so that it can replicate when the host also replicates. Okay, this is what this whole chapter is about. How are you going to get your gene of interest, your desired gene into the genome of a host? Why do you want to put it into the host? Because the host will replicate. And when the host replicates, you will obviously expect your desired gene to replicate as well. Okay, so when this host is replicating, you will obviously look for that host which replicates very fast. Tell me children, which is the most favorite bacteria? Which is our favorite bacteria for your NEET exams? Which is our favorite bacteria for NEET exams? Tell me. E. coli, very good. So mostly we will see that the host which we are going to be referring to would be E. coli. Why? Why E. coli? Why E. coli? Because of the very short time that it takes for replicating itself. How many minutes? Tell me in the chat box, children. Okay, so now whenever replication begins, replication is always going to begin at ORI. What is ORI? ORI stands for origin. Okay, ORI stands for origin. Replication always begins at the origin site. What is origin site? We will come to that. Now, when the very first recombinant DNA was made, okay, it was made by linking a gene, okay. What was that gene all about? I told you, desirable gene, a gene of interest. So what is that gene of interest which we are looking for? We are looking for an antibiotic resistance gene, a gene which is resistant to antibiotics. Okay, so now that antibiotic resistance gene will be taken and it will be put into a plasmid. What is plasmid behaving as, children? Plasmid behaves as vector. Plasmid is the reason how we will be able to get our desired gene into the host. Yes or no? That is how it will go into the host. So, which plasmid are we going to be taking? We will take the plasmid of Salmonella typhimurium. Which is it? Taking the plasmid of Salmonella typhimurium. So, what is our gene of interest? Antibiotic resistance gene. What is our vector coming from? Vector is plasmid from Salmonella typhimurium. Who are the scientists that created the first recombinant DNA? One, Stanley Cohen, and also with him Herbert Boyer. In which year? 1972. So in 1972, Stanley Cohen and Herbert Boyer, okay, they are the ones which created the first recombinant DNA. Now, what do you see over here? First recombinant DNA was created by taking the gene of interest, the plasma from Salmonella typhimurium. Okay, what did they do? They needed to then, what are these gonna be called as? Molecular scissors. They are called as molecular scissors. And molecular scissors can also be called as Molecular scissors can also be called as restriction enzymes. They can be called as restriction enzymes. Excellent. Now, so your gene of interest is the blue one. Okay, you have this blue gene on the whole red colored DNA. Do you want that whole red colored DNA? No. So cut off the part that you want. Cut off using the molecular scissors, and this is what you end up with. That is what you end up with. You can now cut it to insert the gene of interest. Where will this be inserted? Into the plasmid. But the plasmid is complete. So to insert it into the plasmid, we need to cut the plasmid. Okay. So yes, here also the plasmid needs to be cut. Okay. So we see here, plasmid needs to be cut. So if the plasmid is cut over here at a specific location, we see that we will then be able to insert the gene of interest. And now this is going to be stuck together in these two places. Okay, so the gene of interest along with plasmid is going to be stuck together with the help of fevicol. Who is our fevicol over here? DNA ligase. DNA ligase is going to be useful to bring them together, to stick them together. Who are these? Gene of interest and plasmid vector. Clear? Now, this what you have here, this is your recombinant DNA. Then, recombinant DNA needs to be put into host. So, you have your gene of interest, you have your vector, and now you have your host. Who is our host? Most favorite, E. coli. So, now, this is the rDNA, recombinant DNA put inside the host, so that whenever the host will multiply, it is going to be, even your gene of interest has managed to multiply. Am I clear? So, this was cloning of your antibiotic resistance gene. Okay, and then sooner in this chapter, later on, we will find out how to extract all of this. Very good. So, now you've created multiples of them. Now, 3 steps. Everyone, now we are at page number 194 of NCERT. Okay, or rather we have come to page number 195. In page number 195, you can see that you can hence infer that the 3 steps in genetically modifying an organism. What are the 3 steps? 1. You identify the DNA. Okay, what is this going to be? Let me just change the color for us. This is going to be your gene of interest. Then introduce it into a host. Correct? Introduce it into a host. So first this will be made into rDNA and then introduce it into host like E. coli. Then maintain the host and transfer the DNA to its progeny. So the host will multiply. Host will multiply and the DNA will be transferred. So the new progeny will have your desired gene of interest. Am I clear? Now, next paragraph, tools of recombinant DNA technology. Tools of recombinant DNA technology. Come on. There are 3 tools we are going to be focusing on. One is restriction enzyme. Second is cloning vectors. And third is the competent host. So let's begin paragraph by paragraph. We start with restriction enzymes. Okay, restriction enzymes. Which is the year? Tell me which was the year when the first DNA was made. Which is the year when the first recombinant DNA was made? In the chat box, do you all remember? Which is the year when first recombinant DNA was made? First recombinant DNA. Very good, Shruti. First recombinant DNA was 1972. Excellent. Now, the first restriction enzyme, okay, was discovered in 1963. Okay, in 1963. Now, there were, okay, in this year, what did they find? They found that there are 2 enzymes, okay. These 2 enzymes are naturally present in E. coli. And what are these enzymes? What were they capable of doing? Whenever they saw that there is a bacteriophage, here is the bacteriophage. Bacteriophage means it is a virus which has the ability to infect a bacteria. Means whatever its genome is, it is able to insert it into the bacteria so that it can spread the whatever the disease. It can infect the bacteria. That's what you call as a bacteriophage. But there were two enzymes inside the E. coli. They restricted the growth of the bacteriophage. They did not allow the virus to infect it. What were these two enzymes? How did they work? Look, one of the enzymes, two enzymes were there, na? One enzyme, what did they do? It added methyl groups to DNA. It added methyl groups to this viral DNA. And what happened was, E. coli had already there were already enzymes which can break down that DNA, the methyl groups. So there were enzymes which in E. coli which can break down methyl groups. So if methyl group was added to this DNA of the virus, then the DNA will break down. Okay, so that is one way. The other enzyme, what did it do? What did the other enzyme do? The other enzyme was capable of cutting the DNA of the virus. So that enzyme was very useful, and that is the enzyme which we are going to be using as molecular scissors, because it is capable of cutting DNA. And we call these, what are they doing? They are restricting the growth of the bacteriophage. They are restricting the growth of the virus. That's why they are called as restriction enzymes or restriction endonucleases. Restriction enzymes or restriction endonucleases. Is this clear? Why are they called as restriction enzymes? Because they restrict the growth of the virus in the bacteria. And they are how do they restrict the growth? By cutting the DNA. And that's why, because they are capable of cutting the DNA, we call them as molecular scissors. Am I clear? Yes, it did not cut its own DNA. Obviously, it will only work against foreign DNA. Okay. Now, the first restriction endonuclease to be identified in the year 1968, okay, that was 5 years later, was HIND2. And HIND2, the functioning depended on a specific DNA nucleotide sequence. Means that HIND2 is the scissor, but the scissor will have its own conditions. मैं तो बस ऐसे ही काम करूँ. I will only work if these kind of conditions are given to me. Okay, so that's what HIND2 says. So restriction endonucleases, restriction enzymes, also just like all other enzymes, will work only in certain conditions, will work only at certain places. Okay, so even this over here, HIND2 had the ability to cut DNA at a particular, it was able to cut DNA at a particular point. It's like, you go to the coffee shop and you say, I will work only if I sit over there. You give me any other table, I will not work. Give me the table which has the cushion chair, I will work there only. Same way, endonuclease only works at a specific site, and that specific site is a site which has got 6 base pairs. 6 base pairs, nitrogenous base pairs, 6 of them. Okay, so the recognition site for HIND2 is consisting of 6 base pairs. Now, we see here, before we learn what are those base pairs, okay, we see that today there are 900 restriction enzymes, and they have been isolated from over 230 strains of bacteria. So 230 bacteria they took, and they took all the restriction enzyme out of it, and they saw that there were total 900. So imagine, you have to cut something and you got an option of 900 scissors to use. How awesome! 900 scissors you're having to cut something that you have. Okay, so we see here, each of these restriction enzymes, now each of them have their own nakre. They all have their own tantrums. I will work here, I will work here, I will work there, I will work there. Imagine you take a bunch of primary children to the garden, or you take them to the restaurant to have some food. What will you do? Will you give them to choose? You chalo. Whatever you want to order, you can order. You'll go crazy. You'll tell them chup chap se ye khao. So these restriction enzymes, 900 of them, have their own 900 nakhre. That's why we say each have different recognition sequences. Each will say I will work only here. Okay, now talking about the nomenclature of a restriction enzyme, okay, we see here, look, the very first alphabet that's used is the genus, okay, the first letter of the genus. The second and third alphabets is the species of the bacteria from which it has been isolated. So you know that the restriction enzymes come from bacteria. Okay, so which bacteria has it come from? It's come from, let's say this one, eco means E. coli. So E for Escherichia, CO for coli. R is the name of the strain, and the name of that strain which we're working with is RY13. And then the last one, 1, Roman numeral 1, because it is the first enzyme which has been isolated from this specific strain. It is the first enzyme. Okay. All right. Now, next we come to kids. Now we are now at just a quick check with your NCERT. We are now at page 196. Okay, second paragraph. Are you all with me? Are you all with me? Page 196, second paragraph, we all are on. Okay, cool. Now, coming to nucleases. Nucleases means some enzyme which is going to be able to break down. It's an enzyme working on nuclease, means on your nucleic acid, on your DNA. So, it's going to be able to break down nuclease, okay, or the nucleic acid. Now, these are what we call as restriction enzymes. We can also call them as molecular scissors. Okay, they are either going to be able to work on the ends of the DNA, or they will be able to work in the middle part of the DNA. Okay, there are those certain scissors which are only going to be working on ends of DNA. Beach mein they're not able to do anything. But then there are other ones which are only going to work in the middle part. The ones working on the ends, outer sides, exonucleases. The ones working in the middle of the DNA, endonucleases. Clear? So exonucleases is going to cut, it's going to remove nucleotides from the ends of the DNA. Endonucleases is going to cut at specific positions inside the DNA. Okay, now while cutting, okay, they can either I've given you always examples of, you know, some sellotapes are there. When you cut them na, ekdam sat karke nicely they'll cut. But then there's some sticky cheapo sellotapes you get. If you try to cut them with your 2 fingers na, they'll just keep on stringing like a long thread. Hai na? So either you can cut it as a sat karke blunt end, or when you cut it, they're gonna stretch and they'll make a sticky end. Okay, so there are gonna be certain enzymes which cut and make a clean cut. But then there are certain enzymes which are gonna cut, but then they'll have dragging pieces along with it. So you're gonna call them as blunt ends and sticky ends. So these enzymes, when they're cutting, you know, you ever seen those baby scissors? When baby scissors, you try making baby scissors cut plastic. Nahi ho paega. You'll never be able to do it. It will just try to do something and ruin the whole plastic paper. But if you have a sharp scissor, okay, like one of those tailors use, and you try to cut a cloth with it, it will cut it so nicely and so boldly. So that way, in the same way, even when you have to cut DNA, depending on which enzyme you will be working, depending on which molecular scissor you will be using, either it will create a very sharp cut end, or it will be creating strings and it'll just long pieces. So those are the blunt ends. Blunt ends are created when you use the molecular scissor Hind 2. Sticky ends are created when you use the molecular scissor E core 1. Okay. Same thing, Dhanush. Yes, we had done this in class. Now, restriction endonucleases, okay, the restriction endonucleases or only endonucleases, they are going to be looking for what? Now, I've said this in my class several times. Let's say I go to Domino's, I go to Pizza Hut, I go to any restaurant. If my son goes and says, Mama, I'm only going to be sitting on that sofa and then only I'll have my pizza. Same way, even these endonucleases have their own tantrums. They say that we will only be cutting if there is a palindromic sequence. Now, what is a palindromic sequence? Here we go. Malayalam. Malayam. Same way backwards. Malayam. So these are palindromes. Look at over here. This is one more A. GAATTC, GAATTC. So this way and this way, it is the same. That's why this whole sequence which you see highlighted is called as a palindrome. That's what you call as a palindrome. Hi Rutuja, hello Pooja. My God, I see all my kids here. Excellent. Now, as we saw, hind 2 creates blunt ends, right? Hind 2 is able to create blunt ends. So look at this, kids. Clearly it is mentioned how is hind 2 working. Hind 2 is able to cut the DNA bluntly in the middle. So can you see how it has been split? Okay, 3 in the palindromic sequence, 3 goes this side, 3 goes this side. But then when E-core 1 works, okay, that is when it creates sticky ends. Alright, what do we see here? We see that when it works, sticky ends are created. So if we have over here GAATTC, GAATTC, how is it going to work? Something like this. Okay, so we see here that this part will go to one end and this part, the other part I'll color, this part will go to the other end. Is that clear? Sticky ends, and this part over here and this part over here, these are called as the tails. They are complementary to each other, but they have been drawn apart. That's why they are called as complementary tails. Is this clear for everyone? Now there is a diagram in page 196, figure 11.1. Figure 11.1. Everybody come to 11.1 diagram. We will just quickly see that. Okay, 11.1. Over here they are showing one. Let's say this is your gene of interest. This is your gene of interest, and this over here, this part here, imagine this to be your vector. Okay, this is the DNA of your gene of interest. This is your plasmid DNA. Okay, now what do we look at? E-core 1. E-core 1 is going to be cutting, but remember kids, E-core 1 creates sticky ends. Hind 2 creates blunt ends. So also you need to know that the very same molecular scissor you're using to cut the gene of interest, you have to use the same one to cut the plasmid vector. Okay, so we are using E-core 1 to cut both of them, and E-core 1 is going to create sticky ends. Here is one sticky end. Okay, I'll highlight it. Here is one sticky end. Here is the other sticky end. Okay, this is the sticky end of gene of interest. This is the sticky end of the plasmid vector. And now, since we have both of them cut, sticky ends free, now let the sticky ends attach to each other. And how are the sticky ends gonna attach with each other? With the help of hydrogen bonds, because nitrogenous bases are joined with hydrogen bonds. Remember, either 2 bonds or 3 bonds depending on which are the nitrogenous bases. So hydrogen bonds are gonna attach them, and let's write the sequences over here. So it will be again G from the gene of interest, C, T, T, A, and let's write the sequence coming from the vector, that is G, and then C, T, T, A, A. Is this clear? So these are now linked together with the help of hydrogen bonds. Okay? Okay? Eco R1, that's also fine. Alright. Now, this what you have over here is your recombinant DNA. This is what we're going to be calling as the recombinant DNA or rDNA. Okay. Alright. So, as I told you, you need to be cutting the source DNA. The source DNA, the DNA, what is source DNA? What is source DNA? Remember where we got your gene of interest from? That is your source DNA. And the vector plasmid, okay, cutting it with the same enzyme, right? Since the enzyme used here was Eco R1, since the enzyme used here was Eco R1, they both have sticky ends and they can be joined with DNA ligases. Okay, they can be joined with DNA ligases as it is going to be the Fevicol. Joining them together, DNA ligases. Clear? Alright, Shruti? Okay, so yes, this is something, repeat, yeah. Okay, so as it is written in the diagram in page 197, everyone, same diagram explained over here. Here is your antibiotic resistance gene, okay, from your, this is your source DNA, this is your gene of interest, this is source. And this is gene of interest. Okay, gene of interest is your antibiotic resistance gene. That is going to be inserted into the plasmid. Plasmid is taken from which? From Salmonella typhimurium. Then they are cut with the help of restriction enzymes or molecular scissors. And after you cut them, you have your gene of interest. Gene of interest will then be attached. It will then be inserted into the plasmid, and this is your rDNA. Okay, can you see your recombinant DNA molecule is right over here. Then recombinant DNA molecule is inserted into the host. Over there also in your textbook they have given the host same as E. coli, and E. coli is going to replicate, and when it replicates, even the plasmid replicates, even the gene of interest also replicates, and that's what we call, children, as cloning of the antibiotic resistance gene. Clear? Sessions की याद आ गई? Very good. Kids, breaking enzyme, Dhanush, enzyme which is able to break down. That's what it is. Okay, let's continue. Now we come to, now look, now you got, let's say now you've done all this process and now you have your required DNA, but you want to separate and isolate the DNA fragments. How do you do that? By a process known as gel electrophoresis. So, what is done over here? Pay attention, kids. DNA is taken, it is cut with restriction enzymes. You have then fragments of DNA. The fragments of DNA can then be separated. How are you going to separate the fragments of DNA? With the help of gel electrophoresis. Okay, how is gel electrophoresis working? Let's see that. No, Devjit, मैं नहीं भूली. अभी फिलहाल we want to study, and that's all we're doing right now. ध्यान दो इधर. If I start my lecture mode now, you all will start your masti mode. Yes. So now, our negatively charged DNA fragments. Step by step, I have included gel electrophoresis. Everyone, please pay attention here. Okay. DNA fragments are negatively charged. This is something you all know. We're going to put them through an electric field. So obviously, this negatively charged DNA will move towards the positive terminal, right? It will move towards positive. So in this diagram also, if you see, this will be where we will have the negatively charged DNA and this will be the positive terminal. So DNA will obviously, DNA is put inside these wells. What are wells? Khadde. What are wells? What are wells? Potholes. है ना? What are wells? Potholes. Potholes are where you're going to insert the DNA. So DNA is inserted. When an electric current is passed through it, the DNA is going to move through this gel. Which gel is being used? Which gel? Agarose. Agarose gel. It's transparent. It is transparent. The gel is transparent. The DNA is transparent. Everything is transparent. But the DNA is inserted into these wells. And DNA is negatively charged. So, the negatively charged, obviously then the wells will be put in the wells are going to be put in the negatively charged terminal. And when the DNA is put inside these wells, and when you allow charge to go into it, that negatively charged DNA is definitely gonna go ahead and it will go towards the positive terminal, towards the anode, right? But this gel which is there, it's like a sieve, okay? It's like a sieve. So when this sieve is there, obviously, which molecules will move faster? Which molecules are gonna be moving faster, kids? The smaller molecules move faster, the bigger molecules stay behind, they move slower. Okay, the smaller molecules are going to move faster, the bigger molecules are going to stay behind near the wells. That's why you can see clearly in this diagram that these thick bands, okay, I'll just make them red. These thick bands you're seeing here are still moving slow. है ना, मोटा होगा, he's gonna stay over there only. The thin ones, the active ones will start moving very quickly. Same way, the thinner, the smaller, the lighter DNA fragments move quickly towards the anode. The heavier DNA fragments move slowly because they have to pass through that sieve-like gel. Okay, so that automatically is filtering the DNA. So we see here, the negatively charged DNA fragments are put through an electric field. The medium used is agarose gel, and that is a polymer extracted from seaweeds. Okay, the DNA fragments pass through the sieve, okay, because the consistency is like a sieve of the gel. Eventually, what is happening because of the electric current, the DNA fragments are now separating. Okay, they are separating. The smaller fragments move rapidly. Now what? Now we have a gel, okay, completely transparent, but you know that because you have put an electric current into it, the DNA fragments have, separated according to their weight, according to their size. So now what do we do? Now we will stain this whole gel with ethidium bromide. Ethidium bromide, you will stain it and put it under UV radiations. Very important question. What color is ethidium bromide going to appear as under UV light? It is bright orange color. Bright orange color. Very good. Bright orange color. Kids, focus on the subject. Focus on the subject. You've got your exams coming up. Yes, it is bright orange colored. Bands of DNA will be visible by UV radiation. NEET question 2021. Okay, bright orange colored bands. Now, these bands are obviously, you know that now you can see them because they're going to be bright orange in color. And then you're able to separate them. You separate them by cutting them. But then, do you want the gel also? We don't want the gel. We only want the DNA. So now we need to remove the gel. The gel is going to be removed by a process which is going to be called as elution. Elution is removing the gel off. Okay. Removing off the gel. Okay. Then The separated fragments, it's like, you know, you go to someone, you have wrapped them a gift, you give them the gift. Do they want the wrapper? No, they'll take out the wrapper, and then they only want the gift inside. Okay, so separated fragments are visualized now under a microscope, and then those fragments are used for constructing the recombinant DNA. Okay, for constructing the recombinant DNA. Is this clear? This was gel electrophoresis from your textbook page 198. Is this clear, everyone? No, Prince. Very good. कहाँ है मेरे बच्चे? सो गए तुम सब? All of you sleeping? All of you sleeping? I told you don't sit to write notes right now. You'll get these notes in the Telegram channel immediately when our session ends. Right now I want you to James. Right now I want you to pay attention with me and revise with me. So I am looking at my chat box constantly. I need your answers. There's no fun in revision if you're just gonna sit quiet and I'm just gonna give you bhashan. Is that any fun? You tell me. Hi Srishti. Oh, I see my unicorns then. Thank you, James. You're here now? Okay, chalo. Let's start. Now, cloning vectors. Okay, cloning vectors. These are the ones, obviously, you know what a vector is. Vector is the one which we are going to be able to put into the host. Okay, vector is the one which is like how you have mosquitoes. Mosquitoes are behaving as vectors now. That's how malaria comes into your body. Mosquito nahi hota, so there would be no malaria. There'd be no dengue. So let's just go and destroy those mosquitoes. But these cloning vectors are the vectors which are gonna be useful for us. We want them. Okay, because if vector was not existing, how would you ever get your required DNA, okay, into that host? It is vector that's gonna help you do it. So the cloning vectors, these are the vectors which are able to replicate in the bacteria. Okay, they're able to replicate in the bacteria. Now, they are either, you can say, you can use a bacteriophage. What is a bacteriophage? A virus. Or you may use a plasmid coming from bacteria. Okay, either you use a virus or you use plasmid coming from bacteria. Now, what is the difference? If you're gonna make a bacteriophage a vector, it is gonna be able to produce high number of clones per cell. So whenever it multiplies, it will be able to create high number of clones. But then when a plasmid is gonna multiply, it's going to be able to create either 1 to 2 per cell or maximum 15 to 100 per cell. Okay, so we see here, if at all an alien DNA is linked with a bacteriophage or a plasmid, then what? It will replicate. Your alien DNA will be able to replicate whenever these two replicate. Okay, it will replicate with the bacteriophage or with the plasmid, and however many plasmids will be formed, that many your alien DNA will be formed. However many bacteriophages will be formed, that many your alien DNA will be formed. And that's exactly what we want. We want to create multiple copies of your gene of interest. That's your alien DNA, right? So the same number of clones of the alien DNA is then produced. Is that clear, everyone? Yes, I will send my notes as soon as the lecture is done. Don't worry. Now, where have we reached? We have reached page NCERT page number 199. NCERT page number 199. Are you all with me? Oh my god, it's already 3:25 and yes, we'll be able to complete. Will we be able to complete? Are you guys with me? Will we be able to complete? Yes? Okay. quickly. Alright. So you guys know which chapter is there tomorrow? Applications. We're doing applications tomorrow. And tomorrow, yes. Tomorrow we're doing applications. Day after we're doing human health. Human health I will be taking for a little longer time maybe. And tomorrow applications also I'll be taking for a little longer time. Let's complete this today. Okay, Srishti, chalo, let's begin. Let's continue, kids. Pay attention now. Features required to facilitate cloning into a vector. NCERT page 199. Which features are required so that you want in order for the vector to be able to clone? Okay, first of all, it needs an ORI. It needs an origin of replication. What is ORI? 3 points. It is where the replication starts. Okay, second point. Any DNA, if a DNA is put over here at this ORI, it can replicate in the host. Okay, third point, very important point for your MCQs. It is the sequence responsible for controlling the number of linked DNA. However many DNA are linked there, how many are supposed to be made, that is controlled by ORI. Important for your MCQs. Okay. Now, point number 2, we come to selectable marker. What is this going to do, selectable marker? You know what this is? This is like a tag. Okay, you know, you have your sticky notes, and then if you see my NCERT textbook also, you've got these tags over here. Are you able to see that? You have these tags. All these tags here are helping you identify something. Same way, selectable markers help in identifying, and also it helps in taking out. Now if I have marked all these chapters, you know what chapters I have marked? Obviously I will mark the zoology chapters. So this helps me eliminating the non-zoology chapters. It eliminates botany chapters. It selects for me zoology chapters. So it helps in eliminating non-transformants and selecting of transformants. Selectable markers help you take out what you don't want. What is it that you don't want, and what is it that you want? You want your rDNA. You want the DNA which has recombined. You want the DNA with the gene of interest. Do you want those plasmids which don't have the gene of interest? No. Why do we want them? We only want those DNA which has the gene of interest. So you're going to eliminate the non-transformants. You're going to select the transformants. So what is a transformant? It is when the DNA means what? It is when the DNA, your gene of interest, it is then introduced. Obviously it's a recombinant DNA, and when it is put into the host bacteria, that is like E. coli, you will call that as transformation. Okay, so when your recombinant DNA is put into the host, you may call that as transformation. What are the useful selectable markers for E. coli? Antibiotics. Okay, so the genes encoding resistance to antibiotics. Okay, ampicillin, chloramphenicol, tetracycline, kanamycin. Okay, now let's continue. Talking about the third, those are the cloning sites. What is a cloning site? It is the site used in order to link your alien DNA to vector. So if alien DNA has to be put into the vector, where will you put it? You will put it at a cloning site. Okay, and preferably in the whole vector, there should be only a single recognition site. Single. If there are more than one recognition sites, then what happens is several fragments are made, and when you have several fragments, it gets complicated. Life gets complicated. So keep it simple, keep it one only. Okay? Alright. So complete gene cloning it will complicate gene cloning if there are many fragments. If you see these words, you might get confused. When you see restriction sites, recognition sites, they are the same. Okay, then we see here there is ligation of alien DNA. What is ligation? Sticking of alien DNA can be done at a restriction site in one of the two antibiotic resistance genes. Which are the two genes we're talking about? ampicillin resistant gene and tetracycline resistant gene. What is this over here? This is an artificial plasmid which has been created. Okay, this is an artificial plasmid which has been created. So, what do we see over here? Children, please pay attention over here now. Please pay attention. We see here, this is your PBR 322. Correct? PBR 322 is an artificial plasmid created which has two antibiotic resistance genes. Which are they? Ampicillin resistant, tetracycline resistant. Right? Okay. Now, let's say I am going to choose BAM-H1. Okay. Where is BAM-H1? What is BAM-H1? It is the restriction site chosen. And which gene is it located on? It is located on tetracycline resistant gene. It's located on tetracycline resistant gene. So, that BAM-H1 has been cut. A restriction enzyme has been used, and can you see it has been cut? If it has been cut, what just happened over here? It is going to lose the tetracycline resistant property. It will lose it. So what will happen if tetracycline is put here? Tell me children, what will happen now after cutting this gene? If tetracycline antibiotic is put here, what will happen? Definitely the bacteria cannot grow. It cannot grow. If at all it has been cut. Very good, Mihika. If it has been cut at this region, okay, this whole gene you see over here is tetracycline resistant gene. If it has been cut over there, this plasmid no longer is now tetracycline resistant. Okay, and then you insert the alien DNA over there. And this what you have now is your recombinant DNA. But now, the tetracycline resistance will be lost. Clear? Clear everyone? So this is your PBR 322. Alright. You can check this whole out. The same thing I have written here. Okay, the green dots are showing ampicillin, purple dots are showing tetracycline. And if these two genes are intact, then even if these antibiotics are there, this will survive. But the minute something gets cut, okay, in the presence of that antibiotic, it will not grow. So in this example, what has been cut? Tetracycline is cut. So the host will not grow because of which antibiotic? Because of tetracycline. Clear? It has okay, now there is a schedule, you can check it out now. It has completely lost the property of tetracycline resistance. It has been inactivated due to insertion of alien DNA. Is this clear, kids? So that's why your tetracycline resistant gene is helping in selecting. So it is a selectable marker. I hope now that's clear. Okay. And because you have, when you have inserted your alien DNA, it has inactivated your tetracycline resistance gene. We call this as insertional inactivation, because after inserting the alien DNA, your gene has become inactive. So insertional inactivation. Clear everyone? Have you all gone and hit the like button? Do I really need to tell you guys this still? Really? Do we need to tell you this still? Hit the like button. Thank you, Sachin. Alright, now kids, let's come to page number 200. Vectors for cloning genes in plants and animals. For plants, we're going to use Agrobacterium tumeficans. Okay, Agrobacterium tumeficans is able to produce tumor in plants. Okay, it delivers tDNA. It delivers tDNA. So, if a normal plant cell, okay, has tDNA inside it, it will produce tumor. Okay, so that plasmid of this is going to be used because the plasmid is able to transfer into the cell, into the plant cell. But what will we do? We will we will put your gene of interest, we will put it into the tumor gene. Okay, the gene which is able to produce tumor, we'll cut it over there, we'll put our gene of interest. So if we're cutting the gene which will produce the tumor, will it be able to produce tumor now? No. What are we doing over there? We are adding our gene of interest. So now it will be able to put our gene of interest into the plant cell. Okay, we are manipulating this bacteria, and that's why this is your cloning vector. So this is the vector which we use for plants, and the vector which is used for animals is called as a retrovirus. Okay, we usually use retroviruses. Now coming to competent host. Okay, competent host, we're almost there. DNA, you all know, is hydrophilic. So if DNA is hydrophilic, it is not able to cross through cell membranes. You want to put the DNA into the cell, but the DNA being hydrophilic cannot pass through the membranes. So you need to make it competent. You need to make it able to go inside. How will you do it? By treating it with divalent cations like calcium. So by doing this, it's going to increase the efficiency. It's going to create pores in the cell wall. So calcium has now been able to create pores over here. And after the pores have been created, we will put it together with recombinant DNA. What is this? This is your bacteria. Put it together with recombinant DNA. First put it through ice. Then give it a heat shock, 42 degrees Celsius. And then put it back into ice. And once that's done, you will see how the bacteria has gone, the rDNA has now entered into the bacteria. Did you all understand this? Did you all understand this? Understood this? So ice, heat, ice. Clear? This is one way of introducing a recombinant DNA into the host. Okay, but then there are other ways to do that too. Other ways to introduce alien DNA into the host. What are they? If it is an animal cell, the recombinant can be directly injected into the nucleus. If it is a plant cell, then the cells can be, gene gun or biolistics. So what is that? Important for MCQs. Gene gun. What is done over there? Look. The plant cells, it's done on plant cells. Okay, they are bombarded with high velocity microparticles of what? Of gold or tungsten. So you're taking microparticles of gold and tungsten, you're coating it with the DNA, and it's going to be put into the plant cell with a very high velocity. And there is a machine that does that. We call it as a gene gun. Okay, so that is for plant cell to introduce DNA. Otherwise, disarmed pathogen vectors means the vectors which is a pathogen, but they're not going to cause a disease. Okay, because we're disarming them. They will, just like how we saw the Agrobacterium tumeficans. Okay, they're able to create tumor. But when you cut the tumor producing gene, they become disarmed, right? And they can go and transfer they can infect the host. Okay, all right. Now, when we talk about the plant cell is bombarded. Microinjection for animal cells. Yes, microinjection for animal cells, plant cells, gene gun. Alright, write out of your textbooks, beta. Chalo kids, we are near the end. Can we do this quickly? Process of rDNA technology. First, we will isolate the DNA. Okay, then fragments are going to be created using the molecular scissors. Then that small DNA fragments will be made, correct? And how will we get the DNA fragments? By gel electrophoresis. Then once you have the fragments of the DNA, of the DNA gene of interest of the vector, then you will ligate that DNA into the vector by using what? DNA ligase. By using DNA ligase. Then transfer that rDNA into the host. Okay, transfer that recombinant into your host, and then culture the host in a medium at a large scale bioreactors. Okay, and then what do you get? You get the desired product. Okay, then you get the desired product, because plants and animal cells are having different membranes through which the DNA, recombinant DNA, needs to pass through. Cell wall, everything is different. Okay, now very first step, page number 201. First step, isolate the genetic material. So we need, as I told you right now, DNA is enclosed inside the cell membranes. Okay, DNA is enclosed. You can close your chat, just watch the video. DNA is enclosed inside the cell membranes. We need to break open the cell with enzymes. So the DNA basically needs to be penetrated inside the nucleus. So how are we going to be, breaking those cell membranes? Look, by enzymes. Lysozyme is an enzyme used for going into the nucleus of bacteria. Cellulase for going into plant nucleus. Chitinase for going inside fungus. Okay, because that's what their cell walls, that's what their cell membranes are made up of, right? Bacteria, lysozymes are antibacterial. Cellulase and chitinase for breaking down the cell membranes of plant and fungi. You all know that DNA is highly coiled. Yes or no? You all know it is highly coiled, supercoiled, right? Nucleosomes, everything is there. So while you try to insert it inside, you need to make sure you want only the DNA. You don't want anything else. You don't want the all the cell organelles, the carbohydrates, nothing. You don't want anything. So you make sure that everything is broken down. Okay, you break down not only the cell membranes but also all the other parts too. You can break down the RNA by ribonuclease enzyme. Okay, you know that DNA is wound with histone proteins. So to break down those histone proteins, the enzyme used is gonna be protease to break down the histones. So ultimately you will have with you left only and only DNA, because everything else has been broken down with enzymes. And this purified DNA ultimately will be precipitating out when there is addition of ethanol. I think they've written chilled ethanol. Yes kids, chilled. Ma'am, alcohol mein baraf. Chilled ethanol. Clear? Okay. Then next point, page number 202. Chill the ethanol. Obvio. Cutting DNA at specific locations. So this purified DNA is incubated with restriction enzymes. So take DNA, take restriction enzymes. Don't give them any other place to go only. You have to stay with each other and give them optimal conditions to work with. Okay, obviously those enzymes will do its work, right? They will cut the DNA. Now that will be put through gel electrophoresis so that you will get your fragments. And the negatively charged DNA move towards the positive terminal, and the same process is going to be done with the vector also. So you get the gene of interest, you get the vector which has been cut, and then what? The gene of interest, the cut vector will be mixed together, add the enzyme called as ligase, and obviously they will create the recombinant DNA. Is this clear? Is this clear? Clear everyone? Perfect. Now the last points I have not made notes. Let's quickly go through that from your NCERT textbook. Will be PCR. Okay, amplifying your gene of interest. You want to create multiple copies of your gene of interest. How will you do that? With PCR. Okay, so PCR stands for polymerase chain reaction, and over here multiple copies is synthesized in vitro using two sets of primers. Okay kids, what I want to do now is, since I'm having a session, okay, I will be completing the notes of PCR for you and bioreactors, and we will do this the first thing in tomorrow's session. Okay, we will do this first thing in tomorrow's session. I will make the notes and share it with all of you. How's that? I'll see you everyone. I'll see all of you tomorrow. Dhanush. Okay kids, PCR, bioreactors and applications. Everything done tomorrow. Alright. I hope this session was useful for all of you. All of those who are new over here, please I need you to understand how important for your NEET exams the next 1.5-2 months is. So please all of you utilize each and every day very wisely. Okay, Vedantu has a very awesome crash course light which has been there. All of those who might be feeling that this is expensive, some things are expensive for you, then this is just for you. We are offering you 120 recorded interactive sessions, okay, of zoology, botany, physics and chemistry, whatever you require. There is, whenever the sessions are scheduled, there will be proper timings for you to come, and also there will be live class teachers there for you, for supporting you in any of your doubts. Okay, you can improve your rank with your part test and all the assignments which will be given to you. Improve your knowledge, improve your scores, your approach to your MCQs with the help of 90 assignments that are going to be provided. Okay, so the teachers here are Rahul sir for physics, Sudarshan sir for chemistry, Bhumika ma'am for biology. Here are the schedules given. You can go through this. You can take a screenshot of all the schedules, how it has been segregated for you all. Okay, so just have a look through this. It's gonna be extremely useful. Please make sure everything, each and every day of yours is utilized very wisely. Alright, this is only and only for 1800 after discount. When you put in my coupon code 1800 mein, you're going to be able to utilize each and every day very properly, and there will be class teachers live with you for answering all your doubts until your NEET exam. So here is the number. If you have any queries, you can just type this out and give your query in. They will help you out. In order to get that deal at only 18 over here, yeah, in order to get this deal of crash course light, which is only at 2700, apply the coupon code SIPRO. Okay, so you will get a good discount. Alright kids, so I will see you all tomorrow, and tomorrow we complete biotechnology and we start and complete applications. Alright, bye everyone. I will see you soon. Come soon tomorrow.